Hi everyone, welcome back to the final session of the day. It's a great pleasure. Me and Rowan get to present the BSCB student and postdoc medal lecturers. So I hope you've all had a good time in the poster session so far. So we're going to start off with the BSCB postdoctoral research medal. And this was an award which actually was instigated by Gutan Day, who was the previous postdoc rep for BSCB. And it's supposed to be awarded to an early career researcher who's made a major contribution to the UK/Irish cell biology community during their postdoctoral training. This is not only through their amazing research, but also through them showing independence and also a contribution to the science community through outreach and other factors. And so it's a really great pleasure that I can award this to Agat Chan, who is a developmental cell biologist/biophysicist who studied her PhD at the CIRB in Paris and then moved to do a postdoc as an EMBO fellow at the LMCB in London with Eva Pulach. Not only is she an EMBO fellow, but she then moved on to be a Henry Wellcome postdoctoral fellow in the same lab. And alongside this has done a whole load of things for the science community, including being part of the postdoc committee, organizing EMBO lab leadership courses at her institute, setting up early career seminars, all alongside teaching and supervising students. She's going to talk today about her research, which has been elucidating the crosstalk between cell division and cell fate transitions, but I'll let her talk to you about that. So take it away, Agat.
Hi, everybody. I hope you can all see my screen and hear me all right. Okay, awesome. So it's really an immense pleasure for me to give this lecture and to receive this award for a variety of reasons, and one of which is because the BSCB meeting's been my favorite meeting for quite some time. So I'm a little bit sad that I don't get to actually, be there in person and, meet everybody, hang out with friends. But I'm hoping this is gonna happen very soon. So today I'm just gonna talk to you about the main work that I've been doing as a postdoctoral fellow in Eva Pulach's lab at the LMCB. And so my project has really been to elucidate how cell division and cell fate crosstalk. And so, as Alex said, I'm a developmental cell biologist. So fundamentally, the thing that I'm really excited about is understanding how you go from one cell to a completely developed organism. And for that to happen, two things happen at the same time. And the first one is the cells need to divide. So here on the left is an example of a mouse oocyte. On the right is a mouse embryo at E14.5. So you see there's a really huge difference between the two. And the first thing that happens is that cells need to divide in order to just build an organism of a bigger size. And the second thing that needs to happen is that the cells need to actually undergo a series of phase transition so that you get an organism with like exquisite complicated structure and different tissues. And so fundamentally, I'm really interested in how those two things crosstalk. How does that happen? And so I built this project in the context of the mammalian development, and I'm really fascinated by early development in the mouse embryo and how the phase transition really are intertwined with cell proliferation. So this is just to remind you what the development of the mouse looks like. So following fertilization, the embryo is going to undergo a series of cell division, and around the 8-cell stage, the cells are going to start making decisions based on their positions. And following that, the cells are going to undergo a series of phase transition that are going to ultimately lead to them differentiating into different lineages. And so I was really keen in understanding how cell division and cell fate transition crosstalk around the E4.5 blastocyst, which is sort of around just before the implantation stage. And so as a model system for the blastocyst, we can use mouse embryonic stem cells. So we can basically take cells from the embryo and then maintain them as pluripotent cells. So if there are some stem cells aficionado, we use 2I-leaf as a pluripotency media. And in that context, the cells divide and remain pluripotent, but we can also remove the pluripotency media, and the cells are then going to undergo a really interesting phase transition, which is called exit from naive pluripotency, and they're going to reach a stage called formative stage, which is just a stage where they actually can be receptive to differentiation signal, and then become one of the fundamental germ layers. And it's been known for a few years that actually inhibiting cell division inhibits this this particular cell phase transition. And so I was really interested in understanding how. And so this study that I'd shown that had been done in bulk. And so to really understand exactly how cell division and cell phase transition crosstalk, I really wanted to look at single cells and understand what happens. And so to look into that, I performed some single cell tracking and could actually see that cells exit naive pluripotency. So they undergo this phase transition right at the time of cell division. And so I was keen on understanding the dynamics of exit pluripotency after cell division. And I'm just going to go really quickly on that. But one key thing that I discovered is that if I looked at sister cells, so cells that emanate from the same cell division, the difference in time of exit between the sister cell was actually really low compared to the difference in time of exit within the population of cells that are exiting naive pluripotency. And that really suggested that somehow the sister cells remain connected because their dynamics of cell phase transition were tightly correlated. And that actually, I made that discovery not that long ago, and that actually brought me back to some observation that I had done years ago kind of serendipitously in looking at cell stain for alpha tubulin, where I actually saw that cells actually remain connected after cell division by cytoplasmic bridges that contain alpha tubulin. So you can see here on the left, this is a staining of mouse embryonic stem cells stained with tubulin in black. And so highlighted by this pink arrow, you can see bridges that are connecting two sister cells emanating from a cell division. And this is here at the center. It's another way of looking at this. It's an image from one of my colleagues, an SCN image where you can see here two sister cells that are connected by this knife bridge running along another cells. And so what are those bridges that I'm talking about? So the bridges are actually the remnant of the mitotic spindle that becomes trapped within the cytokinetic ring. So the cytokinetic ring progresses, and then the spindle and some proteins that are associated with the center of the spindle are going to remain trapped in those bridges. And later on, the escort machinery is going to be recruited at one side or both of the bridge, and they're going to cut the bridge in a process called abscission, removing the proteins, the central protein part, which is called the midbody. So in just a little bit more detail about what exactly happens, this is a nice image of the spindle. Most of what's going to happen during abscission is actually defined at the center of the spindle. And you don't really have to remember all those molecule names, but basically abscission is in 3 steps. The first one is recruitment of a number of key players at the center of the spindle here, defining where the cytokinetic ring is going to constrict. The second step is the anchoring of some of the proteins at the center, which are going to become the midbody, and a secondary constriction. And the third step is the recruitment of the escort machinery itself, which is going to actually perform abscission, so the cutting of the membrane. And so just a few proteins that are important. This one is called citron kinase. It is a nice marker of the midbody, so you find it at the center of the bridges. ALIX is a protein that is important for recruiting the escort machinery at the bridge and perform abscission. And CHIM4B is the mammalian escort shrew. And so, okay, what I've told you so far is that naive cells, so pluripotent cells, they have a high number of bridges, which you can see here very clearly stained in white. So those are all bridges. And you can see in pink in the middle is the citron kinase I just mentioned. So it marks the center of the bridge where the midbody is. But actually, if you look at cells when they're no longer pluripotent, so when they've exited naive pluripotency, you can see that they no longer have any bridges. And this is quantified here in a little bit more detail. And so bridge maintenance is actually decreased at the exit of naive pluripotency. So how does that work? And so to answer that question, I actually looked at the structure of the bridges by electron microscopy with Ian White at the LMCB. And I could see that something that was very clear to me is that the structure of the bridge was quite different between pluripotent cells and cells that are no longer pluripotent. In that the naive bridges, so pluripotent bridges, were actually much thicker than bridges after cells exit naive pluripotency. And that suggested that what is impaired in pluripotent cell is the capacity to undergo secondary constriction. So what are the consequences of that on actually abscission? And so to look into that, again, I looked at CHIM4B, which is the mammalian escort-3. And I could see that in cells that are no longer pluripotent, they actually can recruit CHIM4B, but when cells are pluripotent, they cannot, as quantified here. So suggesting that the secondary constriction cannot happen when the cells are pluripotent, and that prevents the recruitment of escort-3 and thus the abscission. And so what are the consequences? I've told you that naive cells have long-lasting bridges and they don't perform abscission, but doesn't matter. What's the consequence of that? And so to look into that, I decided to maintain bridges when cells are no longer pluripotent and see the consequences on pluripotency. And to do that, I targeted ALIX, which, as I mentioned, is important to recruit the abscission machinery at the bridge. And so using siRNA against ALIX, I could see that I was able to maintain a high number of bridges when cells exit naive pluripotency compared to control, who actually perform abscission faster and no longer have bridges. And using a functional assay that helps me probe how fast cells exit pluripotency, so how fast the state transition happens. I'm not going to go in detail about this assay, but I could see that cells that have bridges actually exit pluripotency slower. And so that suggests that inhibiting abscission actually impairs exit from naive pluripotency. So how about the opposite? Can we actually accelerate abscission? And what are the consequences of that in pluripotency? And so to look into that, I went for a coarse approach using laser ablation to actually ablate bridges between cells exiting pluripotency. And so here you can see there are two cells here that are connected by a bridge here. You can see this is sertubulin staining the bridge. You can see here nicely in this transmitted light image, this white bulge here in the middle is the midbody. And so you can shoot the laser at the bridge and cut the bridge. And you can also nicely see where the midbody actually goes. And looking at the time of exit from naive pluripotency using a reporter for pluripotency, I could see that when the cells are ablated and do not keep the midbody, so for example, this cell here at the bottom left corner, they actually exit pluripotency faster, suggesting that bridge ablation accelerates exit from naive pluripotency. And so that brings me to my working hypothesis, which is that abscission actually couples cell division to cell fate. So what happens is that when cells are pluripotent, they divide, and at the end of cell division, contrary to a lot of other cell types, they do not sever the connection between sister cells. And this is actually important to maintain pluripotency. However, when exit from pluripotency is triggered, they are able to rewire the abscission machinery, and this allows them to exit naive pluripotency and actually separate the two daughter cells after cell division. And this is crucial to allow this cell fate transition. So of course, there are still a lot of questions that are not answered, which are, for example, how do the cells actually do that? So we have some clues, but the complete mechanism is still to be investigated. Which cells are actually able to modulate the rate of abscission like that, and how do the bridge actually sustain pluripotency? And these are avenues that I'm quite excited to be working on. And to finish, I would like, first of all, to thank the BSCB for this wonderful meeting and this great award. As I said, it's it's really a great pleasure for me. I would like to thank my lab, in particular, Eva, who's been a great mentor and has allowed me to sort of bring my project to the areas I was interested in. Everybody in the lab, and in particular, people here in blue are past members that have been involved in this project or some other that I haven't had time to mention. Everybody at the LMCB, in particular, microscopy facility, that I spent a lot of time bothering for one thing or the other. And my collaborators in Cambridge, all the people I talked to, and in particular the BOEM lab, and I've had an extremely great peer network at the LMCB that has been really important and I think really crucial to me, being able to perform this project and all the activities that I've been doing. My collaborator Edouard in Vienna, and all my funding sources, and you for your attention, and I'm happy to answer any question that you may have.
Thanks, Agatha. That's a really nice talk, actually. So we've already got loads of questions coming in, so I think we've got time to answer at least a few of them. So the first question is, have you looked at other bridge components such as RhoA, anilin, etc., in the pluripotent cells? Do they stay at the bridge until S-core is recruited?
Yeah, so that's a great question. So I'm just right now compiling a list of all the proteins that I need to look at. So no, I've looked at a few components, none of which you've mentioned, none of those that you've mentioned. But yeah, that's absolutely a next question, to understand exactly which proteins are involved in maintaining those long-lasting bridges. So which proteins are not where we expect them to be, and what's the dynamics of them being actually recruited to allow this phase transition. But right now, I can't answer that. I don't know.
Yeah. So the next question is, amazing. Love an escort talk. Did you investigate Chimp4C localization in pluripotent state? Is Aurora B persistently active?
So I haven't looked at Chimp4C yet. I have looked at Aurora, and I've only looked at its localization, so Aurora and Phospho-Aurora, and it looks normal compared to what you would expect in a HeLa cell in terms of localization. Like, I can't say much about activity just yet, but same, it's also on the list.
Yeah. What happens when sister cells divide? Are multiple cells bound together?
No, so that's something that is quite interesting. I need to mention, I think, that those long-lasting bridges, they also exist in the mouse embryo up to the at least 32 cell stage. And what they see and what I see is consistent, is that the bridge actually is dismantled prior to the next division. So in the embryo, I think it is just prior to the division. In stem cells, it looks a bit more heterogeneous, but in that sense, you only have like two cells bound together. It's only one bridge. I have to mention that there are other cell types in which, cells can actually undergo the next division while still being attached. And so you can have like cysts that are formed, or cells with like a lot of bridges between them. But that's not the case in any of the animal embryos that I can think of that maintain bridges.
Okay. You may already have answered this question, but someone's asked, for how long do the sister cells stay connected, and do daughter cells also have the bridges?
So the length of the bridge is actually, so as I said, in embryos, it seems that they're as long as the cell cycle, and so it's pretty stereotypic, even though I don't have numbers. In embryonic stem cells, it is more variable, which may or may not be linked to the variability of the cell cycle, but basically I think the median is around maybe 6 hours, and it can go up to like 10 hours, which is basically the duration of a cell cycle in mouse embryonic stem cells. So it's basically 12 hours, the cell cycle. So it can go up to 10 hours. Okay.
Someone's also asked, are mid-bodies reabsorbed by cells, and does this impact cell fate?
Okay, you're cutting off a little bit, but I think the question was, is the mid-body reabsorbed by cells, and does it impact fate? So the live imaging I've performed doesn't really allow me to answer that, but it's very clear that the meat body can be reabsorbed by a variety of cell type from some of my images and a lot of people's work. It has been shown that it can impact fate in a variety of cell type, including stem cells, not by me, I have to say. It is not clear whether it signals when it's in the bridge, it signals when it's released from the bridge, or it signals when it's absorbed by the cells or on the cells. In HeLa cells, it seems that the meat body can signal proliferation from being attached to the membrane of the cell. In the case of stem cell, it is not clear exactly what happens.
Okay, hopefully you can hear me now. The final question that's been asked here is, can you speculate why this abscission doesn't occur? Do you think that it could feed into Notch signaling?
Why it doesn't happen in pluripotent cells? I don't know about Notch signaling. I don't think so, but I don't know enough about Notch signaling to say for sure. Speculating about why it doesn't happen, my best bet at the moment is that there is some abscission checkpoint. There are a few abscission checkpoints that are known, and I think one of them must be activated to prevent, which I think relates to a previous question about aurora. I'm not sure it is via aurora, but probably one of the proteins that is upstream of the escort complex is not being recruited, or is being recruited, preventing recruitment of chimp. But I can't say which yet. But that's basically the next step of what I want to look at.
I just want to end on one question from me, actually, which is actually probably really naive, but they seem to exit at different time points. Does this exit rate actually affect their fate? So if they exit really quickly, do they go on to be mesenchymal cells? Do you know what I mean? Or
yeah, absolutely. So I've had some preliminary data on that, which it was a bit of a different setup. I basically sorted the cell by cell cycle and then triggered them to go to RC4 lineage. And so the first cell that exit, which are the ones that basically are triggered to exit when they're in G1, seem to be quite efficient at forming mesoderm, but not so efficient at forming ectoderm. And so that I don't know why at all, but it's also I mean, there are some data on this kind of stuff, like how the cell cycle actually controls the fate in human embryonic stem cells. So that's quite consistent.
Okay, thanks, Agatha. I just want to remind anyone, if there's any more questions, please come to the social we have at 6 to 7, where you can meet the medal winners, and Agatha will be there. And I'll now hand over to Rowan.
Thank you.
Thanks. Thank you, Alex. I'm just going to share my screen. There we go. So I'm here to introduce Flora Paldi, who is the first recipient of the BSCB's new RAF medal, which is an award that was created by Joyce Yu, who's the previous PhD rep of the BSCB, to recognise PhD students who've made an outstanding contribution to UK Ireland cell biology. Candidates for this award were assessed on their research excellence based on a project summary and community engagement, such as conferences, public engagement, community outreach, or scientific advocacy. So Flora was awarded a first-class degree in molecular genetics at the University of Edinburgh, during which time her undergraduate research project contributed to a second author publication. And Flora was subsequently accepted to undertake a Wellcome-funded 4-year PhD at the Wellcome Centre for Cell Biology at the University of Edinburgh, where she joined the lab of Professor Adele Marston to investigate the structural organisation of pericentromeres in budding yeast. Flora will be sharing this research with you today, which has been published in Nature subsequently. Flora is now a postdoc with Giacomo Cavalli in Montpellier, where she'll no doubt continue to make further outstanding contributions to the field of cell biology. Congratulations on this well-deserved award, Flora. I'll now hand over to you to share your research with us.
Okay. So hi, everybody. Thanks for the introduction. I'd like to thank, first of all, the BSCB committee for awarding me this medal. It's a tremendous honor, and I'm very, very happy to be here and tell you about my PhD work. So as you heard earlier this afternoon from Adal's presentation, the Marston Lab is interested in understanding how accurate chromosome segregation is achieved during cell division. Now, my project focuses on the mitotic metaphase stage of the cell cycle. And just a quick reminder for all of you, in metaphase, DNA replication has been already finished, and the two replicated copies of chromosomes called sister chromatids are held together by a ring-shaped protein complex called cohesin. In metaphase, spindle microtubules that emanate from opposite spindle poles capture sister chromatids through their kinetochores, which are large protein complexes that assemble on centromeric DNA. And actually, once this stage, so-called biorientation, is established, tension is generated on chromosomes by the pulling forces of microtubules, and this tension signals proper microtubule kinetochore attachments and allows cell cycle to proceed to anaphase. My research interest is the pericentromere, which is a short chromosomal region flanking the centromere. And the pericentromere is highly enriched in cohesin, where cohesin is thought to establish a sort of preferred kinetochore geometry for capture by microtubules. Accordingly, this pericentromeric cohesin enrichment is important for chromosome segregation, and cells that actually lack it exhibit increased rates of chromosome loss. So at the beginning of my PhD, I set out to answer two main questions. First, what was the structural organization of the pericentromeres? And second, what were its implications for chromosome segregation? To start addressing these questions, first I wanted to map how pericentromeric cohesin localizes both before and after biorientation. And to do this, I synchronize cells through a genetic arrest in metaphase, which I'll be calling the tension condition. And also in a parallel culture, I did the same genetic arrest, but I also treated cells with microtubule depolymerizing drugs, which I'll be calling the no tension condition. And these conditions recapitulate chromosomal states before and after biorientation has been achieved. So in these synchronous cultures, I went ahead and did calibrated cohesin ChIP-seq. And here is an example of that, pericentromere 4. And what we could see was this high density of cohesin inside the pericentromere in the absence of tension, which decreased in the presence of tension. Now, this tension-sensitive behavior of pericentromeric cohesin was documented before, but what I noticed was that this tension-sensitive cohesin domain was flanked by these prominent tension-insensitive cohesin peaks. And just to be clear, I'll be calling these chromosomal regions and these cohesin peaks as the borders of the pericentromere. So next, we wanted to know where cohesin at the pericentromere borders is coming from. This is important because current models posit that cohesin accumulation sites on chromosomes are distinct from its loading sites. So in the case of pericentromeric cohesin enrichment, is a result of centromeric cohesin loading, where the cohesin loader complex associates with a kinetochore subcomplex, loads cohesin on chromosomes, and from the centromeric side, cohesin then bidirectionally translocates and accumulates throughout the pericentromere. To answer the question whether or not cohesin at these border regions is coming from the centromeric loading side, we used a mutant where we abolish the centromeric cohesin loading pathway by the deletion of the CHL4 kinetochore subunit. Importantly, arm cohesion remains intact in these mutants. So once again, I did a calibrated cohesin ChIP-seq, and that's chromosome 4. So I hope it's easy to appreciate that in the absence of centromeric cohesin loading, we see a decreased occupancy of cohesin both throughout the pericentromere and at these border regions, which tells us that at least partially cohesin at the pericentromere borders is coming from the centromeric loading site. Another interesting feature that we found based on these ChIP-seq experiments was that actually these tension-insensitive cohesin peaks at pericentromere borders always form in between convergently transcribed genes on all 16 budding yeast chromosomes. And this will be important later for the talk. So we thought we mapped the pericentromere cohesin domains and pericentromere borders, but we also wanted to confirm then that these findings are relevant to the behavior of pericentromeres in vivo. And to this end, we made use of a phenomenon that is often referred to as centromere breathing, and it involves the transient separation of sister chromatids prior to anaphase. This you can see on this image here, where I labeled centromere 1 with GFP. And in the absence of tension and due to the tight cohesion of sister chromatids, we can see only one GFP dot per cell. Whereas when biorientation is established, this tension that is exerted by the microtubules transiently splits the two GFP foci on the sister chromatids, and we see two GFP dots. So we wanted to use this phenomenon to confirm borders, and we hypothesized that if the borders identified by ChIP-seq are also important in setting boundaries to this region of separation, then if I label chromosomes outside the pericentromere borders, then I'll be seeing one GFP dot per cell, whereas labels inside the pericentromere borders should separate at high frequency into two dots. So I took two chromosomes, chromosome 1 and chromosome 3, which had a relatively small and a relatively large pericentromere respectively, and I labeled them at varying distances from the centromere. After that, I synchronized cells in metaphase, and I counted how many cells had 1 versus 2 dots inside them. The trend was pretty clear on both chromosomes. So inside the pericentromere, we had a high separation ratio of the GFP markers, whereas the markers stayed mostly together on the outside of the pericentromere. So this was supporting our hypothesis. But what I like to highlight especially is this 12 kb distance, which is falling outside the pericentromere on chromosome 1, but inside the pericentromere on chromosome 3, and showed this huge separation, huge difference in separation ratio on the two chromosomes. So this told us that the region of separation indeed does scale with the pericentromere size that we see on the that we define based on the ChIP-seq data, and that the region of separation doesn't only depend on distance from the centromere. Now, at this point, we started to think that it might be really the structure of the pericentromere that is important for biorientation, and that upon biorientation, pericentromere gets restructured, as it's obvious from the microscopy that sister centromeres move apart from one another. So to understand what was the conformation in this region, I set out to do a HI-C, which is a technique that enables us to elucidate chromosome conformation inside cells. So first, I'll show you the no tension condition. And if you're not familiar with reading HI-C maps, I'm just going to highlight a couple of features. So this map is centered on centromeres. And these darker signals on the left and the right side of the centromere indicate high interaction frequency regions, whereas this lighter color indicates lower interaction frequency. So this sort of pattern is actually consistent with the separate looping of the left and the right side of the pericentromere that are in incomplete isolation from one another. Then in the tension condition, we have seen a striking change in pericentromere structure. So these high interaction regions on the left and the right side of the centromere were completely eliminated, but these low frequency interactions immediately flanking the centromere were preserved. And this is consistent with a V shape where the centromere is at the apex. Now we took this one step further because of course we wanted to understand what was the role of cohesin in setting this structure at the pericentromere. So I did HI-C in the same mutant that I showed you before, where through the deletion of CHL4, we abolish the centromeric cohesin loading pathway. And as you can see, there is a decrease in the sharpness of this pattern that we see in the wild type cells. And this indicates a sort of loosening of the structure and an expansion of the pericentromere. Now, these maps here are average maps. So we piled up the signal across all 16 centromeres to get these nice clear interaction maps. But this pattern was actually beautifully visible on some of the individual chromosomes as well. So that's chromosome 10. And here you can see in the absence of tension, the separate looping of the left and the right side of the pericentromere, the V shape coming up in the presence of tension, and the loss of the sharpness of this pattern when CHL4 is deleted. But what was even more striking when we aligned the high C maps with the corresponding cheap seek tracks, and what we've seen that this tension-sensitive cohesin domain is actually perfectly aligning with this region that undergoes this massive conformational change when biorientation is achieved, and is flanked by these prominent tension-insensitive cohesin peaks. So at this point, we really wanted to know what was the significance of these convergent gene sites at pericentromere borders, because it's been long known that transcription is able to alter the chromosomal localization of cohesin by simply pushing cohesin rings along the chromosome. So we hypothesized that if it was convergent transcription that restricting cohesin in these border regions, then if I rearrange genes into a tandem orientation at pericentromere borders, then cohesin might just slide further along the chromosome arm. So I indeed made this strain. I just flipped the border genes into a tandem orientation, and I'll call this strain the reoriented strain. So I went ahead and did cohesin cheap-seq, and the result was extremely clear. So in the absence of the convergent gene sites, we completely lost the cohesin accumulation at the original border positions. And instead, we started to see this kind of low level of accumulation a bit further down the chromosome arm. Now, this could be thought of as essentially an expansion in size of the pericentromere. And if so, I should be able to detect this with the GFP dot assay. So I went back to microscopy and I labeled the wild type and the reoriented chromosome 4 both inside and outside the pericentromere and scored the dots for separation. For the wild type chromosome 4, the results were similar to that of chromosome 1 and 3, where we've seen high level of separation inside the pericentromere and low level of separation outside. But in the case of the reoriented chromosome, we completely lost this difference between the inside and the outside of the pericentromere. So this really told us that actually cohesin that accumulates at these convergent gene sites is functional in holding sister chromatids together, and it's resisting the tension that is exerted by the pulling forces of microtubules prior to anaphase. To further show that it's due to the convergent gene sites and nothing else, I did a rescue experiment where I inserted short model genes to sort of recreate an artificial convergent gene site in the reoriented chromosome. And although it didn't lead to a partial rescue, it restored the difference between the inside and the outside markers in terms of separation. So we really proved that that it's the presence of convergent genes that is functional in accumulating cohesin and restricting the region of separation. I of course did a high C in the reoriented chromosome, and just to say it briefly, we dramatically change the pericentromere structure in the absence of these convergent genes, and we start to make asymmetric contacts between the left and the right side of the centromere, which is completely absent in the wild type strain. So, because at the beginning I told you that this pericentromeric cohesin enrichment is thought to establish a preferred kinetophore geometry for capture by microtubules, we wanted to know if this alteration in structure also leads to problems with biorientation. So to this end, I did the same assay as Adele explained you earlier this afternoon, where we arrest cells genetically in metaphase in the presence of nocodazole. Then we wash the drug out, letting the microtubule reforms. And over time, we can follow the separation of GFP markers, which is a readout for biorientation efficiency. What I could consistently see that actually these reoriented, the reoriented chromosome showed consistent delay in and reduced frequency of biorientation compared to its wild-type counterpart, indicating that these pericentromere borders indeed promote biorientation. Now, the lack of two dot separation is also due to incorrect attachments. So we thought that maybe in the absence of proper structure, this reoriented chromosome is making more incorrect attachments than the wild-type counterpart. And if so, then it would mean that, that it relies more on the error correction machinery, which actually severs these incorrect attachments, giving the cell another chance to biorientate chromosomes. And to test this idea, I crossed the reoriented chromosome into a hypomorphic Aurora B background, which actually has a compromised error correction system. And when we looked at cell viability following nocodazole treatment, we've seen this marked loss of viability, which was actually specific to the reoriented chromosome in this sensitized background, indicating that in the absence of proper pericentromere structure, cells indeed rely more on the error correction machinery. And now, just to sum up, I'd like to finish with this model of how we think this happens in the cell cycle. So we think that cohesin loading starts at the centromere, but instead of its canonical function in holding sister chromatids together, this pericentromere cohesin is actually making intrachromosomal contacts and extrudes these loops in the pericentromere. This loop extrusion then continues until these convergent gene sites at pericentromere borders are encountered, and the back-to-back geometry is established at the kinetochores, which facilitates proper microtubule kinetochore attachments. Then when biorientation is achieved, the pulling forces of microtubules simply expand these pericentromeric loops, and the cohesin that was forming the loops can just slide off chromosomes. Whereas cohesin that resides at the pericentromere border regions remains functional in holding sister chromatids together and resists the pulling forces of microtubules. So altogether, this is how we think that targeted cohesin loading at the centromere and the linear arrangement of transcriptional units in the pericentromere eventually sets up a proper chromosome structure that makes chromosomes competent for segregation. And just really quickly, I'd like to thank everyone who was involved in this project, especially Adele for an amazing PhD experience, Bonnie, who did the initial experiments for this project. I had a great deal of help from our bioinformatics core, especially Dan. And I'd like to also acknowledge our collaborators at the University of Sussex, especially Steffi, who taught me how to do Hi-C. And finally, I'd like to thank the University of Edinburgh for my entire higher education, and also welcome for funding our center, our lab, as well as my PhD. And thank you for your attention.
Thank you, Flora. That was an incredible talk. I think we've got time for a couple of questions. Got any coming through? So first one is, do you know if the pericentric bio-oriented genes are actively transcribed? Are there other genes closer to the centromere?
Yeah, this is a very good question. And so first things first, they are transcribed, all of them, and they are normally not cell cycle regulated, so constitutively transcribed. For the second part of the question, it's not always the first convergent gene pair that is closest to the centromere. In most chromosomes it is, but it's not always. But what we could essentially figure out that at the border convergent genes that accumulate cohesin, normally transcription that goes towards the centromere is higher than transcription that goes away. So these seems to be a defining feature.
I'll ask a quick question. So are the only things controlling it are the cohesin that's there and the genes in the actual chromosomes, or are there any other proteins that are involved in controlling the size of that separation?
So we mostly think it's cohesin. We looked at condensin, which also localizes in the pericentromere. But based on the Hi-C data, it didn't seem to have any effect on the pericentromere structure.
Thank you very much. for lack of time, if anybody does have any, there are some more questions coming up in the chat, please come to the breakout session that's happening directly after this, and Flora will be happy to answer them then, I'm sure. All that's left for me to say is thank you very much for joining us today, and to all of the speakers today for their presentations. We hope you've enjoyed everything. Final day tomorrow, so make sure you all turn up for that as well. Please remember that the poster and oral comm prizes will be announced tomorrow during the closing session. Directly after this, there are a couple of events going on. So there's the Meet the Medal Winners session, which Flora and Agat will both be at, and the other medal winners will be in a separate breakout room as well. And there's also a round table that you can attend on how to succeed as a minority scientist and how to grow diversity in science. Thank you both very much for some incredible talks, and we'll see you in a little bit.